All right, everyone, what's up? How's it going? It's a nice night in Pennsylvania tonight. It's a good night to watch some crazies. So, before we get started, I have just a little bit of background for this documentary. I have never seen it. I know of it, but I've never seen it. But when I was looking for it in preparation for tonight, to actually like stream it and react to it, I found this. So this is the Children's Health Defense.tv, Robert F. Kennedy Jr.'s organization. This is promoting this 7-part virus denial documentary. This is Mary Holland interviewing who are these people? The filmmaker of the Viral Delusion, and Mary Holland is the president and general counsel of Children's Health Defense. Yeah, she interviewed these guys, she platformed them, and this is a documentary that promotes the idea that viruses don't exist and cause disease. Now, I tried to look at the Children's Health Defense actual website for this video, and I've had to go to the Wayback Machine because this link wasn't working. But when I go back to the Wayback Machine and I look at this archive, which is from September 25th, 2022, it pops up for a minute, but then I get an error message. So I don't know what that's about, but I did find it on Rumble. And yeah.
Right there, davidraznick.com. And I think you told us that there are decades with Duisberg's book about the AIDS virus, of Dr. Peter Duisberg's book.
I want you to hear this.
David Raznick can show us that Robert F. Kennedy Jr.'s book has some parallels to Peter Duisberg's book about the AIDS virus.
So, yeah, that clip I think shows it. She's platforming virus deniers. She's sympathetic to virus deniers because Robert F. Kennedy Jr.'s book spends a significant fraction of its pages denying the fact that HIV causes AIDS. Wild stuff. All right, so tonight I'll be reacting to this documentary that Robert F. Kennedy Jr. platformed. I have it up here, and I'm just going to play it, and periodically I'm going to pause it and give my commentary on it. But hope you guys enjoy this reaction, and let's get into it. I'm expecting a masterful cinematic opening on par with The Lion King or Saving Private Ryan. So let's get started.
In February, I took a trip to California, and on the West Coast airports in California, I saw people wearing masks, and I said, this has to be serious. I've never seen this before. And that's when I just started saying, okay, let's figure out what's going on. I'm going to start at the very beginning. I'm going to look at the papers that discovered the virus and, take it wherever it leads me. And I never expected to find that those papers didn't actually show any virus.
Oh, yeah, they did. Right away.
I was really surprised that the experimental techniques that they were using, it couldn't possibly show a new organism. It was totally the wrong experiment to do. At first, I was just confused. I'm like, I must be missing something.
Yeah, we're really hitting the ground running, guns blazing with this.
Far off. It was just shocking, and I had to really talk to other people and look at their work and look at the historical context before I could fully appreciate what was going on.
With any virus, to show that it exists, you need to separate it entirely from any biological material.
And the studies, and the most important studies, claiming that they have proven SARS-CoV-2, they haven't purified the particles. And without purification, And finally, isolation. You cannot.
What? All right. That was weird. All right, so basically all they're saying now is the classic virus denier trope of confusing what isolation is and what virology is. What's this say? A growing number of doctors and scientists, blah, blah, blah. Okay. All right. Yeah, this is totally garbage so far.
When somebody says, a virologist says, we have isolated a virus, I thought, as does everybody, that they took...
Quick note, this guy thinks that the heart doesn't pump blood. He thinks it's not a pump. Movie volume low. Okay, I'll turn it up a little bit.
100 people or 500 people who are sick in the same way, right? They have a runny nose or a cough or pneumonia, and then you do steps, like you take their secretions.
That's true. From zero, it can only get better.
And then you centrifuge it, and then you have a band that has just those viruses, right? That's what everybody thinks they did. And then you have
some people did that.
Tell people the steps, so some other lab could repeat the steps. That's how you do it in science.
Yeah, that's
done.
done.
Everybody says, yes, this thing that we have is pure SARS-CoV-2 coronavirus.
Mm-hmm. Yep, have that.
That's what every human being understands by the word isolation, and that's what every doctor and human being thinks happened in this case, right? Except it didn't happen once.
Not exactly. God, the volume on this thing is all over the place. Alright, while it's going through that cinematic intro, I'm just going to mute it. So the way we isolate virus is to take fluid or sample from a patient and then filter it or clean it up in some way so that you take out all the bacteria and big things like fungi that can also be in that sample, human cells, and then you put it onto a culture of cells that are permissible to that virus. And then those cells grow that virus. You can measure the growth of that virus. You can measure the production of its proteins, the replication of its genome, and you can propagate that virus over many cell cultures. That's the basics of it. It doesn't have to be a sucrose gradient like he's describing. All right, let's see what they're saying.
Again, written out with medical history. It's a scientific fight that goes back more than 100 years.
Yes, they are actually saying that viruses. They are actually saying that viruses don't exist.
But its modern incarnation began as a disagreement in 1985 between on-the-ground AIDS researchers and none other than a young medical bureaucrat named Anthony Fauci.
It was declared on April HIV and AIDS thing, April 23rd, 1984, before any scientist or anybody else in the world knew there was an AIDS with a capital AIDS, contagious infectious disease.
There's the AIDS
nobody
denial.
knew. No scientific paper was published or anything about it. It was just declared on that day, and it became government policy, and it's still government policy to this day. That set the stage. Not only that, it set the stage with having a disease with a virus that has never been obtained from a human being. This HIV thing, the only place that I know of that it exists is in the laboratory.
Nope.
Nobody has ever published obtaining or isolating or even seeing HIV in a human being.
Yeah, they have.
The young Fauci had thrust into the mainstream.
We literally know the structure of the HIV virus. We have cryo-EM structures of it at very high resolution, and we know that it infects CD4 T cells, which are depleted in AIDS patients. That's all been demonstrated in humans and in laboratory models.
Ideas of cancer researcher Robert Gallo that a particle he had claimed to have discovered, the HIV virus, was the cause of AIDS, that this virus could be sexually transmitted, and thus AIDS was at the very edge of
What is this
becoming a
B-roll?
new global plague. While much of the world rushed to congratulate the NIH, a small group of doctors and scientists pointed out
This is
that the
weird.
declaration had no serious scientific backing, and that the so-called HIV
What's this B-roll
virus
from?
had,
What is
in
this?
reality, never even
Just
been proven to
Death
exist.
playing bowling?
I was here living in San Francisco. I had my own little biotech companies. So I wanted to work on this like everybody else in the Bay Area wanted to work on AIDS. We wanted to put our skills to work to try to cure it. Be heroes. Be carried on people's shoulders, like all young scientists. We talked about AIDS from the very beginning when it popped up in the 80s, even before it had a name. And then here it was in 84, it's a virus and everything. Oh, good. I'm a chemist. I'm going to make inhibitors
What
to
is
kill
this
this
bird chirping?
retrovirus.
Can they?
And
Oh, they're
the other
interviewing
people
him
working here
outside,
and there.
right,
And then
okay.
by 87, I've been starting to question this stuff. 85. But between 85 and 87, I started talking about the problems with this whole notion that AIDS is contagious, caused by a transmissible infectious agent.
Oh, okay.
All those anomalies, But one of the first things that made me suspicious back, I think it was 84, 85 for sure, about this HIV business was, how does this virus know that you're gay and I'm supposed to cause Kaposi sarcoma in you,
Oh my God.
and not in these IV drug using white men? I give them TB or something like that, I mean, that was
Jesus Christ. He doesn't understand. Kaposi sarcoma wasn't strictly for gay people. He knows that, right? Surely he has to know that. Okay, for those who don't know, HIV was first noticed in the gay community because HIV, what it does is it depletes your CD4 T cells, wipes out your T cell immunity, and then things like the virus that causes Kaposi sarcoma can cause Kaposi sarcoma. And it was unusual to see that in young men. But there were a significant number of gay men in New York City and San Francisco who were getting this. Ow, fuck. Yeah. That's how it was first noticed. But it wasn't exclusive to that community. It also was being observed in heterosexual people, in drug users, in people who got transfusions of blood that was infected with HIV. Wow. He thinks Kaposi sarcoma was only in gay people.
It's one of my first suspicions, And also, how does that little virus know not to cross the street? Like in the Castro area. Stay live here where the gay guys are, where they have all these AIDS diseases, but it doesn't go over here in this other part of town. It's a virus that knows your zip code, whether you're gay or straight, white or black, or whether you're rich or poor. That's one of the first things, those discrepancies goes back. By 1987, I realized this whole shoot match with contagious sexually transmitted HIV was not tenable. In fact, it was crap.
The debate quickly blew up when the leading virologist in the U.S., Peter Duesberg, accepted the government's claim that what was pictured...
He was not a leading virologist in the U.S., no. Oh, damn it. Sorry.
...waged behind the scenes. As the debate shot through, the side accepted the government's claim that what was pictured under the microscope was a so-called virus, but published a paper explaining in 19 clear points why this particle could not be the cause of AIDS.
But
it's dormant.
It was.
To begin with, it's dormant when you die from it, it's dormant when you suffer from it. There's no report in the literature describing the virus ever to be active in a patient.
Yeah, there is.
It was a silent war waged behind the scenes.
You can literally...
As the debate shot through the scientific community, the critics were soon joined by Carrie Mullis, the Nobel Prize-winning inventor of the PCR process.
Yep, who also denied that HIV causes AIDS.
Up our grant from the NIH to work on that, and I had to write it. And so the first line of that was HIV is the probable cause of AIDS. And I wrote that, and then I said, well, I need a paper, some kind of scientific paper, to reference that statement, because when you make a scientific statement like that, that's like a fact. So you need to
plenty
say,
of papers
here's
like
how
that.
come I know that. Right? You put a little 1 if it's the first statement you've made, and then you put down at the bottom of the paper you have a 1, and you say, here's a paper by somebody that describes why that statement's true.
Yes, thank you, Carrie. We know.
So I said,
we know.
I said, well, what's that? I don't know. Let me think about what is that paper? Who do I go to for that?
Robert Gallo, Luc Montagnier, several others.
That company, and they said, no, you don't have to reference. I said, I have to reference that because I don't know where that came from. How do I know that? And it turned out that nobody knew it. There wasn't a scientific reference, like a paper that somebody has submitted with like experimental data in it.
Yes, there
and
is.
like logical discussion, and said, here's how come we know that HIV is the probable cause of AIDS. There was nothing out there like that. Nothing.
Mullis insisted his technique was being misused to claim it could find viruses and diagnose disease, when it could do neither.
PCR is separate from that. It's just a process that's used to make a whole lot of something out of something. That's what it is. It's not. It doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything like that.
Right.
Mullis became Fauci's most prominent.
Yeah, PCR does not tell you that you're sick. It does not tell you that whatever it's detecting is an infectious agent, but it does detect HIV. And separate experiments demonstrated quite clearly that HIV does cause AIDS. If he wanted papers to cite, let's see. He could have cited papers like these. This is written by Luc Montagnier and Robert Gallo, two people that first isolated and characterized HIV. It tells the story of how it was determined to cause AIDS. Pretty straightforward. He could have talked to them, but he didn't. Oh well.
Critic, and soon was joined by numerous other leading scientists, but it was a debate that was kept almost...
Yes, they are saying that hepatitis C does not exist either. Yeah, these people think that 0 viruses exist.
...entirely out of the media.
To say that Anthony Fauci controls the media is a real understatement.
Celia Farber, great HIV
Peter was
AIDS
going to be
denier.
on one of the talk shows. I don't know if it was CNN or if it was one of the big. I think it was actually one of the networks, and they were just going to do straightforward journalism. Oh, Dr. Duisberg, you are an esteemed retrovirologist.
This is weird B-roll. They're showing footage from the previous interview
but what do you have to
with
say?
another interview taped over it.
Flew him to New York. He was in his hotel room and about to go downstairs to get picked up by the car, and phone rings. Oh, sorry, Dr. Duisberg, we have to cancel the interview. And he says he literally turned on the TV at the appointed time, and Tony Fauci was sitting there talking about the latest numbers and the latest scare and how. So they must have had such a surveillance capacity even then, and/or spies and moles and all this kind of stuff. So they knew. They absolutely knew.
Does someone like just slam a book and no one cut it out?
The argument came to its most devastating culmination over Fauci's prescription that those who had tested positive for the HIV virus, using Carrie Mullis's PCR test, should take a repurposed chemotherapy drug known as AZT to save their lives, while Fauci's detractors argued that the drug was a known and extraordinarily deadly poison, which would...
Okay, before we go on, they're taking a lot of footage, like just directly from another terrible documentary called House of Numbers. If anyone is interested in seeing a complete debunk of this House of Numbers documentary that they're heavily using footage from, go to Miles Power's YouTube channel. He did an entire debunk of the whole documentary in multiple parts. It's really good.
...was killing the very people who thought they were saving themselves.
No, it wasn't.
It was an argument over a new disease, but not a new argument. 30 years earlier, a number of prominent doctors had spoken out forcefully that polio, too, had been wildly misdiagnosed.
What?
And that the devastation of childhood paralysis was much more likely due to the introduction of new insecticides than to the sudden and capricious rise of the virus. They, too, were silenced by a growing medical bureaucracy
No, okay, so they're claiming here that polio is caused by DDT and not poliovirus. No. Back then, people weren't sure what exactly caused polio. Some people thought that polio was transmitted by flies. So what would happen was, after a polio outbreak would strike a town, they would spray DDT to try to kill the flies. Right? They would spray DDT after the polio outbreak got into a city. It wasn't the DDT. My God. We literally know what receptor on cells polio uses to infect them. We can take that receptor, put it into cells that it can't infect, and suddenly that virus can infect the cells. Conversely, we can take that receptor out of cells that it can infect, and it can't infect those cells anymore. I mean, it's wild. Virologists have done so much work that these people just completely ignore.
Headquartered at NIH, under the influence of the now dominant post-World War II pharmaceutical companies. Even earlier, a silent war had been waged in the medical community over smallpox, which had come to a head when the very doctor who had been selected to write the Encyclopedia Britannica entry for smallpox vaccination set about to look at the research and found, to his shock, that vaccinations had clearly not been the reason smallpox had declined. Though considered the most prominent British doctor of his day, his entry was deleted, his career attacked, and his research written out of orthodox medical history. The thrust of these doctors' and scientists' arguments is that a number of profoundly unscientific
Yeah, that's a good point. Polio definitely predates DDT.
Used to place the blame
it's a pretty old virus.
On the concept of tiny, unseeable entities named viruses.
We can see them and detect them.
Be found in sick patients. When the real causes of disease, such as smallpox, polio, and AIDS, were industrial contaminants, toxins that emerged from poor living conditions, malnutrition, and new pharmaceutical products that were destroying the body in untold
Oh my God, tell that to people thousands of years ago.
Despite their efforts, a rapidly growing edifice of bureaucracy was forming, built on the foundation of virus theory, and the idea of viruses as the cause of disease permeating the culture so profoundly that slowly most doctors came to believe in it. The debate has reemerged today, as a growing group of doctors and scientists are speaking out against the experiments that form the justification for the SARS-CoV-2 pandemic.
Oh, God.
Their argument is that while thousands of experiments have been done regarding the SARS-CoV-2 virus, they are built on an unscientific foundation that is, in reality, pseudoscience.
No.
Today, medical authorities have placed virology center stage in claiming it has the answers to human sickness.
Not all
the
human
rapidly
sickness.
mutating deadly SARS-CoV-2 virus must be controlled through urgently necessary population-wide medical experiments and the rewriting of the world's long-standing social contracts. But the argument of their detractors is that this story has become transparently false, that the experiments to justify these extraordinary actions, when looked at closely, are unscientific and
COVID was not the flu.
Have not prevented, but caused death, disease, and mayhem on an enormous scale, and must be brought to an immediate end before more.
How much of this footage do you think was predating COVID?
The mainstream view is well known, but the arguments of its detractors have once again been
going with the died suddenly route.
This is their story.
Why do they keep putting B-roll of other people talking over a voiceover?
When the pandemic began, on the pronouncements that a new, deadly, and highly transmissible virus had been found. The claims were based on a single paper out of a laboratory in Wuhan.
It started with a single paper that was then corroborated by a large body of
doctors and
data.
Scientists were examining this
this paper.
And the subsequent ones in detail. Here's what they found.
The first thing I did, I actually went to look for the first Chinese study from the CDC, CCDC, the Chinese Center for Disease Control, published under the name Zhu and others, which was the first supposed isolation of the SARS-CoV-2.
Yep.
And of course, immediately found
that was the first paper that isolated it.
discrepancies and and a methodology that didn't isolate anything.
They
Such as
found some people with atypical pneumonia, which is not uncommon. Every year we see lots of atypical pneumonia.
It was
That's what we
atypical,
often call,
unexplained
flu or
pneumonia.
virus, because we don't really know what's causing it. But every winter you see this. So I was like, well, they see a few cases of atypical pneumonia. That's not unusual.
That was not explained by bacteria or known viruses.
They were all at this outdoor food market, which didn't have the best sanitary conditions. So I'm like,
Right,
if
did
people
not have
got
good
sick
sanitary
from there,
conditions.
I would think some kind of food poisoning, but they never even came up with that idea. They said,
Food poisoning
it
doesn't
must be
cause
something like
pneumonia.
SARS. And it was just, why would they come to that conclusion? And then this was also the weird thing, because if you really do have a new disease, right, you would want to take a good sample of cases to study, like 1,000 people, or at least 100. They had less than 10. Why would they only study such a tiny number of people? And I think they only actually took samples
because
for
you're not going to
these tissue
study
culture experiments
1,000 biological
and then all of a
samples
sudden they're assuming it's like some
for a
deadly
new virus.
plague.
So-called isolation or discovery of this virus.
You don't
Done
need
in
to
a
do
single
that.
individual, a single human being. All right. There were 44 cases of pneumonia in December, I guess it was.
So that paper, that first paper that isolated SARS-CoV-2, literally sequenced the SARS-CoV-2 genome. Ask any virus denier how that genome exists when SARS-CoV-2 does not exist. Ask them, where do the proteins that genome produces come from, if not from a virus? They will not be able to answer. Dr. Kaufman, this guy who is talking and calmly denying reality, he's a psychiatrist. He doesn't have a medical degree. He's not a scientist. He's a psychiatrist. Yeah, but a full genome of SARS-CoV-2, these people will deny it by misunderstanding how next-gen sequencing works. But maybe later in this video, I'll show you a paper that they cannot refute. Let's just keep listening for now.
And the World Health Organization office there in China cataloged these 44 people of pneumonia that they had no explanation for. And this is in a country that has about 1 million pneumonias every year.
Right, they had no explanation for it. Usually when people come into a hospital and they have pneumonia, you can diagnose it by doing bacterial cultures or detecting a virus that is known to cause pneumonia in them. But when there's no reasonable explanation, then they have to figure out what's causing the pneumonia so they can treat the people. And when they can't find any known pathogen causing the pneumonia, they look for a new pathogen. And they found one. Because situations like this don't happen every day. There's a very intricate system in China and other places that are designed to quickly detect unexplained diseases, just like that. And that's what happened. It did its job.
All right. What is so important about 44 cases? Like they don't know what's causing it. Lots of things can cause pneumonia. Chemicals can. Malnutrition can. Drugs can.
Yeah, but that's not, those things aren't
old
infectious.
Can. Being in a hospital can. Chronic diseases. I mean, there's all kinds of things that can cause pneumonia.
Being in a hospital can cause pneumonia. What in a hospital do you think is causing pneumonia? Is it an infectious agent? Maybe. Mayhaps. Whomst might be able to guess what might be causing pneumonia in a hospital setting? I wonder. Lots of sick people in one area. Are they spreading pathogens between each other? Who knows?
And with this one guy, okay, so now they have this one guy in a country of over 1 billion people.
So
it wasn't one guy.
What I found was that they were taking the liquid from patients, and they were maybe centrifuging them or filtering them, but
right.
I made a calculation that in the 15 micrograms that they were using to do the test, they were still like about 30 billion particles, virus-like particles, of which 85 to 95% of human origin, human genetic origin, and they would take this very heterogeneous material and put it into a culture, and then they would say that that was the isolated virus, which of course didn't make any sense.
That's not all they would do.
Samples from the patient to try to find a virus, but they don't look in that fluid from the patient for the virus.
Yeah, they do.
I'm like, well, why not? That's the source of it. Instead, I see they mix it in this foreign cell culture, monkey cells. I'm like, why would they
Doesn't have
do
to
that?
be
It
monkey
doesn't
cells.
make any sense.
All right, let's see. So, Vero E6 cells, a eukaryotic cell that is very permissible to viruses, easy to grow viruses in. 10% fetal bovine serum. That's growth factors. That lets cells divide. You literally need that in order to grow cells in culture. You do that regardless of whether or not you're looking for viruses. Let's see, what else? 1X-L-glutamine, that's an amino acid. Penicillin and streptomycin, that's to kill bacteria, which you don't want in a culture because it'll kill your cells. These people just don't know what cell culture is or what the ingredients do.
They basically combined the words virus and isolation together and created a new definition for that term.
Nope.
And this is why it's so confusing. So only the
confusing to
scientists
you.
or doctors who actually take the time to read the paper and read the methods
Which
are
are
going
experts.
to understand that there's no isolation going on.
There is.
Everyone else who just reads the title of the paper, they'll just, oh, well, yeah, they said it right here. They said isolated.
They don't actually isolate viruses. They
Yeah,
just
they do.
say that they isolate viruses. You know, if you think of something like a bacterial species, and it is possible to
this
isolate
should be good.
bacteria, as in just get the bacterial cells separate.
Yeah, okay. Before I see this, how do you isolate bacterial cells? What do you do? Let's see.
Separate them out from everything else, and then
How do you do that?
analyze them, work out what their composition is, their genetics, their structure, work out what the bacteria, how they behave, what they can do, etc. And I think most people think that with viruses they're doing the same thing.
We are. Yeah, he didn't explain how they isolate bacteria. You take bacteria and you put it on food that it can grow on, either a liquid broth or a solid broth, or a solid medium, which we call agar, and then the bacteria grows. Viruses also need food to grow. Food meaning a host that it can actually use to replicate itself. So you put bacteria on agar to grow it, and you put viruses on cells to grow it. It's the exact same concept. I wonder if he believes in the molecular tools used to study and characterize bacteria, because it's the same molecular tools that we use to study and characterize viruses.
Except that with viruses, when they use the word isolation, it doesn't mean that they've physically separated and purified the particles.
Yeah, they have.
It means something totally different. So that's the first deception.
So the question is, why don't they do it? Why don't they show us this virus from a sick person?
We can.
Not only in 100 people, but in one person.
Yeah, we can.
I mean,
We do that
so I don't
literally
know the answer
all
to
the time.
that, but what they say is, well, there's not enough of these virus particles in a sick person to find them.
Oh
Which
man.
is odd, because if there's not enough viruses in a sick person to find them,
oh boy,
what's the theory that they're going to make us all sick and kill us all?
Hold that thought, Mr. Heart
So
is not a
that's
pump.
obviously
I'll
ridiculous.
find you a paper.
In the virology papers that form the foundation of the pandemic, indeed, the term isolation is used in an entirely different way from its ordinary usage. What the papers mean by the term isolation is that the nasal or throat swab has been isolated from the patient, not that the virus itself has been isolated from the nasal swab. Without isolating the hypothetical virus, these doctors and scientists argue that no independent variable can be properly studied or characterized. It is the first step in what is considered the bedrock of both the scientific method and the founding principles of microbiology, known as Koch's postulates. Through FOIA requests, an activist named Christine Massey has received confirmation.
Oh hey, I debated her. Anyway, let's see. Yeah, okay. So here's one paper. There are lots of papers like this of taking samples directly from a patient and looking at it in an electron microscope and diagnosing viruses. This technique is used in tandem with other diagnostic techniques to confirm the exact identity of the virus, but certain viruses have very distinct morphologies that we can see with an electron microscope, and we can see those particles in samples directly taken from patients. No cell culture needed. There are lots of papers like this. This is just one example that I found quickly. Anyway, I debated her, this person, Christine Massey, and when I asked her if she knows where proteins come from, she said, what was it? She said, "I don't know, Dan. I'm not a chemist." She doesn't know where proteins come from. And she wants to say that viruses, which are made up of proteins and genetic material, don't exist. But they're going to talk about her FOIA requests, which are frivolous requests for very specific things, worded in a specific way so that the response is, we don't have that, and then she parades it as a victory. For example, she'll ask in a FOIA request, do you have any SARS-CoV-2 that was purified without using cells? And they'll say, no, obviously. And then she says, see, I win. Because she has no idea what she's talking about.
From more than 150 government institutions, including the CDC and NIH, that they have no record of a virus named SARS-CoV-2 ever having been isolated directly from a patient. Instead, these institutions point to scientific papers which use three methods for claiming they have discovered the existence of the virus, without ever having isolated a virus itself. The first of these is known as the cytopathic effect.
People have sent me in this past year, let me just take a guess, 50 papers called The Isolation of SARS-CoV-2. Now, maybe 48, but, you know, it's a lot. And
just get an exact number.
You know, both the normal virology community and the sort of alternative community sends me paper after paper saying, here's how they isolated. Here's a paper showing the isolation of this virus.
Here it is.
Every single one did it the exact same way.
That's SARS-CoV-2.
They take snot and they inoculate it onto tissue culture,
proteins.
usually monkey kidney cells, which are called vero cells.
It doesn't have to be monkey kidney cells.
Put it in this recipe of the toxic cell culture with, you know,
the first paper to
and
isolate SARS-CoV-2
specifically toxic to the
actually used human cells.
Culture. So when they use kidney...
Sorry, shush for a minute. The first paper to isolate SARS-CoV-2 actually used human cells directly taken from a patient's airways. So if we can inoculate if we can take the same sample and inoculate vero cells, human airway cells, or HEC cells, and get the same virus replicating, how is it something that the cells are producing when they die, which they're not doing in just regular cell culture? No virus denier has a good answer to that.
Cells, they use antibiotics like streptomycin, gentamicin, and amphotericin.
All of which kill bacteria and fungi, not eukaryotic cells. These people need to understand that literally when we're just growing cells, when we're just growing them just to maintain them, like we don't have to be using them for an experiment, we have antibiotics and antibiotics in the culture. Because when you open the flask, even if it's under a sterile hood, there's a risk of contamination. And we don't want contamination because that ruins the cell culture and it ruins your experiment. So we grow cells continuously in the presence of antibiotics and antibiotics. They don't die in the presence of those things. It's only if you add the virus do they exhibit cytopathic effects. But let's see what they say. The antibiotics and antibiotics do.
Which are all specifically toxic to the kidneys. They starve the culture, and then they add other biological materials like bovine calf serum.
That's always in the
like
media.
Fetal bovine serum, rather, like from calves.
Fetal bovine serum is literally always in the cell culture. You need it in order for the cells to replicate. Without it, they don't have growth factors. They don't replicate very well. This has been empirically determined over decades. And try it. If you take FBS out of your cell culture, good luck growing them.
In the womb, their blood. And they can show these, what they call cytopathic effects, which is damage to the cells from this every single time. And then when cells are damaged, they make particles like exosomes and apoptotic bodies and other types of particles that don't even have names.
Mm-hmm.
And they can just point to whatever particle they want and say, there's the virus.
Nope.
So it's very convenient.
Exosomes and apoptotic bodies don't have specific genomes and proteins that are unique to
viruses. The way
viruses.
that they prove, inverted commas, SARS-CoV-2 exists is through stressing cells in cell cultures.
Nope.
Vero cell cultures. They stress them with antibiotics, all sorts of mechanisms, and then they see things bud, or they see...
Nope. Okay. This that they show, this is a experimental plate where cells have been infected with SARS-CoV-2. This is a control plate. Different outcomes.
Cilia on the cells stop beating, and so they interpret those as virally caused, or as viruses budding, or
right, in combination with all the other experiments they do.
The human cells, they stop beating, so the cell must have died. But it could be due to all sorts of things. Very, very
no, not really.
Uncontrolled experiments, one-shot experiments.
The cytopathic effects are just non-specific effects that occur in a test tube when you put various cell cultures, and then they usually start with the cell culture line, which is highly abnormal. So you don't start with a healthy cell line because they find that they don't seem to do very much in the test tube. So typically they take something weird like a Vero, which is a monkey kidney cell. And
they keep saying monkey kidney cells, but they never mention the fact that they use multiple different cell lines for the same virus.
For your experiment, because these cell lines are highly reactive, meaning that they have a high propensity to die and break up in a test tube and produce various exosomes and things that the virologists are looking for.
No.
So in this regard,
that is not why we use Vero cells. We use Vero cells because they're very permissive to viruses because they don't produce interferon. That's why. They don't readily die. They're a cell culture, they're a staple cell line in fields other than just virology because they grow really well. They're easy to grow in cell culture. They don't die. That's the whole point. Oh my God. Mark Bailey, man. What a loon. And yeah, these people make a lot of money by lying. The one webinar that me and my buddy Senstrand crashed and just totally embarrassed these guys in, what was it? I calculated that they must have made at least like, I'm not going to remember it now. I put it in my video on it, but it was thousands of dollars, over $10,000 if I remember right, or around there. But don't quote me on that. They make a lot of money doing this.
Say that all of those effects are due to the presence of a disease-causing virus. But the problem with that is that you can get the same effects by putting other things in the test tube with the cell culture. So you don't need to put a virus in there. It's just the process itself, which stresses.
But only a virus will replicate and you'll be able to propagate the effects.
What John and I call double deception. Firstly, saying that they've isolated a virus when they haven't. And secondly, attributing effects that we can see anyway without the presence of a virus but saying that
there
it's
are
specifically
definitely
to a
unique
virus.
effects that a virus
And this
can
is
cause,
the basis
and
of
unique
modern
results
virology,
in certain
basically.
tests that a virus
And
causes.
It's simply fraudulent. It simply doesn't establish A, that there's a virus, and B, that it's able to cause disease.
So
yeah, on this episode of
here's
Ancient
an
Aliens.
article that was in Viruses May 2020. They're talking about now they have a better name for just debris coming from the breakdown of our tissues. They call it extracellular vesicles and exosomes.
No.
But that means when we get
Exosomes.
poisoned or sick,
Exosomes are extremely distinct from viruses. There is an entire field of biology that studies exosomes. They are not viruses. Viruses have proteins that your own cells cannot make because we cannot find those genes in your cell's genome. This is a great pause game, by the way. Look at that beautiful face. Viruses have proteins that can only come from their own unique genome.
then we break down into these particles. And the question of this article was, can we
this
tell
might as well
the
be
difference
Ancient
between
Aliens.
this breakdown versus something coming from outside, right?
Yeah, that guy does
So
have
he
a very
says,
convincing
quote,
accent.
However, to
I'll
date,
give him
a
that.
reliable method that can guarantee a separation from extracellular vesicles and exosomes from viruses, quote, does not exist.
Yeah, it does.
We can't tell the difference.
That's not true. Actually, we can use affinity purification to purify viral particles from things like exosomes. Because viruses will have particular proteins on their surface or part of their capsid that we can use to target in an affinity column where you have a column with a bunch of antibodies that are very specific to those viral proteins on it. And you can use that column to purify just those viral particles. It does exist. He just doesn't understand it. And we can also use sequencing methods to isolate the viruses away from all the other genetic material. This guy's just this guy is a complete wacko. I cannot tell you how wacky this dude is.
And essentially what that means is all of these genetic pieces that we're calling viruses are just the breaking down of our own tissue.
No, they're not.
If they really were going to do some kind of tissue culture experiment for something that caused pneumonia, why wouldn't they put it into lung cells, human lung cell culture?
They literally did. That was literally... Oh, wait, okay. Jesus Christ, I have to find this now. Let me find that first isolation paper. Okay, so here it is. This is the first isolation paper. What did they do? They isolated it with human airway epithelial cells taken directly from a patient. This is not a cell line that they bought. They took it directly from a patient. It's literally what they did. My God. And he acts like he actually read these papers. Give me a break.
What it supposedly infects.
One group did it on various human cell lines, and none of them grew. And so the conclusion should have been this SARS-CoV-2 virus doesn't affect human tissue.
It literally was first isolated in human cells. It's ridiculous. What cells did they use? Adenocarcinoma, which is a cancer cell line, human liver cells, HUH7, that's a human liver cell cancer, human liver cancer cell line, HEK cells, it'll replicate in HEK cells. Yeah, so they didn't use that many different cell types. And it's just because it doesn't exhibit cytopathic effects in other cell lines, it doesn't mean that It's not actually replicating in the other cell lines.
Okay, thank you very much.
And look, it even says right here, they showed modest viral replication incompatible with SARS-CoV-2 infection. But it did replicate. So it can infect them, just not well. But it infects human airway epithelial cells. They don't talk about that.
Let's move on. You just proved it.
That's not what they did.
Well, that's what they did, and that's what they found. But they came to the conclusion.
Let's see.
They don't say. They just say it worked better with Vero cells. Now, here's the interesting thing. It worked better with Vero cells, which are kidney cells, instead of human lung cells.
No, it didn't. There were no lung cells. Oh, Jesus Christ. If you take human airway epithelial cells, it replicates just fine and causes cytopathic effects. These people have not read the papers that they deny.
Because the antibiotic that they used to sterilize it happens to be particularly poisonous to kidney tissue as opposed to lung tissue.
No.
So it's not surprising.
Only at concentrations that are well above what is used in cell culture.
That the kidney tissue broke down better than the lung tissue. If they used an antibiotic that was specifically toxic to the lungs, they would probably have found out that it does grow on human lung cells.
Nope. They did
again,
find out that
when
it
you
grows
hear
on
this,
lung
you
cells.
think, no, it can't be that stupid. And
I feel the same way about
actually
you,
know what to
Thomas.
say to that for most people. All I can tell you is that's what they did. That's the conclusion they came to.
Read their papers, you'll see that they have
Stefan
disproven
Lanka,
themselves
favorite
with their
loon.
own writing, right? Of course, you realize they are completely anti-scientific because they are not questioning the base, the historical base, and what they are doing. It's just a belief system. They're doing it in faith, and they came up with the conclusion. It's easy to check this, very easy. You see only they work with structures from cell cultures and never ever were able to visualize something in the blood and you know,
we literally
from
have.
your mouth, from urine, from
We
sperm,
literally
from all
have.
other kind of liquid or tissue in the body. Never ever.
Literally
I
have.
absolutely can tell you after a year of looking, there has not been one case in the published literature where a virologist or a scientist took somebody who was sick, did the normal isolation procedures, and said, here is this virus.
To prove the point, Stefan Lanka conducted the same experiment.
Oh, this will be good. This will be good. I've already debunked these experiments, but oh man. All right, hold on. Let me get this paper up for you real quick. So this paper is one where people directly observed SARS-CoV-2 particles in postmortem tissue. Yep, they detected the virus multiple ways, so this is electron microscopy, right? Electron microscopy showed the characteristic crown-shaped SARS-CoV-2. And then they also used biochemical techniques to confirm that this was, in fact, a SARS-CoV-2 particle. There it is. No cell culture needed. Directly from a patient. Okay, so what Stefan Lanka is going to show here is pretty terrible cell culture technique. That's basically what it boils down to.
Without adding any material to a monkey kidney cell culture, along with the antibiotics used in the SARS-CoV-2 experiments. His results were the same. Even without adding any so-called viral material, he was able to see the cytopathic effects in the cell culture.
And how long did
I
he
spoke
grow
with a
those
number of
cells?
virologists in making this.
He grew the cells for an absurdly long time until they essentially died of starvation. That's what he did. You don't. A normal scientist who knows what they're doing in cell culture does not grow cells to the point of starvation when they're doing any virological experiments. Never.
Documentary. None would appear on camera, except for Stefan Lange. But when pressed about the issue of isolation, all of them tried to move the conversation to the two other ways that the scientific papers have claimed proof of the existence of SARS-CoV-2.
Here we go. Genome
The
sequencing.
electron photographs and the genomics.
There we go. Called it.
All sorts of pictures, purporting to be of this virus, and they were put out by the government and other sources. They're published in
By the
magazines,
government.
newspapers, and they're floating all around. And so many of these images were just completely made up, an artist's rendering.
Completely made
What the
up.
heck is this? Why are they trying to pass this off? People thought these things were.
All right, just to be extremely clear, here's a paper I showed briefly earlier. This is a cryo-electron micrograph of the SARS-CoV-2 spike protein. It's imaged from these particles, which are viral envelopes with spike protein sticking out from them. If you get enough of these SARS-CoV-2 particles in your images, you can take hundreds of thousands or tens of thousands of pictures of these spike proteins and average them all together, and you get a consensus structure, which is this, the spike protein. You can then see the actual amino acid side chains on the spike protein. These spike protein amino acids match exactly what we would expect from the genome which we sequence. These are pictures of SARS-CoV-2. Kaufman was shown this paper by Senstrand during one of his webinars, and he dismissed it because it uses vero cells to grow them. But he has no explanation for how this protein got there when vero cells don't have the gene to make this protein. Zero explanation for it. He just ignores it and moves on his little grift. But let's see what he has to say, and let's get to the genomics part, because that'll be fun.
Real. And then I saw the pictures in the paper, in the Jew paper, and they show these images where, cells, from the monkey kidney culture, and then these all different kinds of little particles, and then they just put a little arrow next to a particle. They like the way it looks, and they say, "Oh, there it is. That's the virus."
Because it's consistent with what a
what
coronavirus
the heck
looks
is
like.
that? How could they recognize it? Just by sight. They have
Because
so many sources.
It has a
of
characteristic
particles
shape.
in this toxic cell
And
culture soup.
they combine it with
Exosomes
other
are
techniques
particles
as
that
well.
all cells make. They come off the membrane.
I mean, look, these look totally different. This is a very regular structure of spike proteins around the viral envelope. This has little. Looks like a blebbing. Blebbing is it looks like the particle is bubbling. It's what a cell does when it's going through apoptosis. This doesn't look like a very regular organized SARS-CoV-2 structure.
Same exact way that they say virus particles do, and they are the
It's
same
not
exact
exactly the same.
size and shape as they say virus particles are, and they are made of the same thing that they say viral particles are, which is they have a membrane with proteins on it, and they have genetic material inside. And the genetic material is of a variety of the same exact types that they say viruses have, which include, DNA, double-stranded, single-stranded, mitochondrial DNA, RNA,
Viruses don't have
all those
mitochondrial
things
DNA.
are reported to be in both exosomes and viruses.
Yes, they.
So I said to myself, they created a recipe in the cell culture to make exosomes because they added antibiotics. And if you look up studies,
There
you'll
are
find
always
that
antibiotics
induce
in cell
exosomes.
culture.
So
Pretty much
they're
always.
creating exosomes. And they're showing particles that look like exosomes. So how do they know they're not?
Because of
There's
biochemistry
no
and
chemical test
genomics
that they
and
did.
techniques like
they couldn't because they
cryo-EM.
didn't identify anything first to develop a chemical test, right? So they're just going by sight.
No.
And the thing is, you can't tell the difference of any of these things by sight. It's just made up.
You literally can't.
And I realized that. I'm like, this is not scientific at all. They
Says
show
the
a
psychiatrist.
picture, of cells and various particles, and there's just an arrow to these nondescript brown particles that they just say, oh, those are the virus, even though there's nothing distinct about them.
Yes, there is.
At all. And they don't even have the spike protein knobs that are characteristic on these images in this paper. So they're basically just showing you particles of dying cells and saying, oh, these ones, they're the coronaviruses. But they have no proof. They didn't purify any particles directly from the patient. They didn't do any chemical analysis.
Everybody thinks you see
No chemical
pictures
analysis.
of viruses from sick
Oh my God.
I thought that. And then somebody asked me, well, can you show me a study that shows that? Well, I don't really know. Well, why do you think it? Well, everybody thinks it. Let's take one example. The first paper from Australia that isolated and has a picture of a SARS-CoV-2 virus outside of China.
Mm-hmm.
So that's what people see. They see this particle with spiky things off the side, which are the spike proteins, little dots, and they say that's the SARS-CoV-2 virus. And that's all
That's not all
most
we
people
do.
and most doctors know. There's the virus. How did they get that? They took fluid from one person, mixed it with their amniotic fluid and bovine serum and horse serum and amphotericin B and gentamicin and put it in a monkey kidney cell and it broke down, and then they got these particles. And that's what they're picturing. But interesting.
And here are viral particles imaged directly from patient samples. This is another good one. I'll link this in the chat because this is a nice one to have on hand for whenever these virus deniers say exactly this. These are negative stained electron microscopy images taken directly from patient samples. No cell culture needed. Yep. Got influenza virus. We've got polio virus. We've got adenovirus. Lots of different viruses here. Yeah. That's it.
When you read how they did it, and I'll read it and then explain it, they say, quote, electron micrographs of section Vero cells showed cytoplasmic membrane-bound vesicles containing coronavirus particles. See box 5b. Following several failures to recover virions with the characteristic fringe of surface spike proteins, it was found that adding trypsin to the cell culture medium immediately improved virion morphology. Now, let's dissect this a little. Virion means an individual virus. So they did the Vero cells, that's the kidney tissue.
Yep.
They found membrane-bound vesicles. They found budding out of the cell. They found these little pieces, particles of genetic material. That's like when you blow up the house, you occasionally get pieces of paper sticking to the walls. That doesn't show whether they're going out or going in, or they're just breaking down. So you see them stuck together. Containing coronavirus particles. There were several attempts, and they didn't look like the ones with the spike protein.
Right, and why was that?
So they didn't find anything that looked like a coronavirus. And then they added trypsin to the medium.
Which
trypsin
is
a pancreatic enzyme, protein digesting enzyme.
Protease.
That then ate away the outside of the protein coating on the virus. And then, lo and behold, it looked like it had spikes on it. And so that's the coronavirus.
It helped release aggregates.
There's a lot of analogies.
That's what you're looking for, Thomas.
The one I came up with was It's like you
because in
have
a human
box
body
of wood.
you have
You
enzymes
say, I think there's
that
wood
aren't necessarily
shaped
present
like stars in that box.
cell culture.
And you look through it and you don't see any. So you call your carpenter friend and he cuts out the edges of the box of wood, and then you say, see, there's the star. And most people would say, wait a minute, that's nuts. Because obviously there wasn't anything that looked like that until you put something that digested the outside to make it look like it has spikes on it.
Made it easier to recover individual virions, which actually look like a coronavirus. Rather
then
than
they also say that
aggregates.
they characterized the viral sequence.
Here we
They
go.
didn't characterize the viral sequence.
Yes, they did.
They made a computer model of a theoretical genome sequence.
That's not how
of a
next-gen
theoretical
sequencing works.
So then they did this massive, what I call a dragnet, for all of the RNA. They bound and determined to find a virus as a cause for this guy. So they did this dragnet for all of this RNA, millions of little strands of RNA in this person, using technology that's called metatranscriptomics.
They isolated RNA, yes. And then they
one of these
sequenced
gene things is technology-driven stuff, where they can look at all the RNA, all the DNA, sequence it, amplify it, trillions of fold of things. All this technology is technology-driven, not science-driven. So I got this person's lung sample of his lung. They do all this stuff, have all this fancy equipment. And they zero down to get all these signals. Then they have a computer that stitches.
This dude looks like he's about to start a cult. He literally looks like he's about to start a cult. My God.
All these little segments, these little fragments of RNA together, out of millions and millions and billions of these things. All right, and they came up with a sequence, and then they decided that they had discovered this virus, even though they never touched the virus at all. And they said that was the cause of this guy's pneumonia.
The
So they
genome is
take
literally the
this,
virus.
As I said, this unpurified mess. So they end up with this vat of literally billions of pieces of genetic material
Yep.
in this stew, right?
Yep.
So that's the cell culture. So they have all this broken down genetic material. Now, you would then think, because this is called an unbiased genome calculation, sequencing,
Okay.
that
So
they look in that brew and say, which is the, what genome do we see? But they actually don't see anything like that. What they do is they don't see a whole genome. So they chop it up into little bits.
Oh, so what the heck? I just paused it on a wow. Shutterstock. Okay, I'm not gonna let that distract me. I'm just gonna leave it paused there. They left a Shutterstock frame in there. Quality videographers here. Okay, so this concept, right? If they truly believed that they could not get a viral genome out of a complex mixture of multiple genomes, then how do you believe that crime labs can separate my genetic profile from someone else's, or human genetic profile from animal genetic profile? These people would literally, if they truly believe this, they need to throw out all of forensic science. Any DNA evidence in a court would have to be thrown out. They would have to do that. But that's not reality, because when you can sequence all of the genetic material in a sample, you can compare it to references and determine what's what. That's why if you mixed my DNA with my dog's DNA, you could pull my DNA and my dog's DNA out of that sample and sequence us both independently. Is wild. Yeah, my buddy Senstrand, when he debated Saeed Qureshi on my channel, asked him, so if I can throw plant DNA and human DNA in a blender, can I tell the difference between the two? And he said yes. and then he immediately had an oh shit moment and tried to backtrack that. It was hilarious. But anyway, let's move on. I'll get to the paper I want to show in a minute.
So some of them are 2,000 base pair, some are 10,000, some are 6, some of them are whatever. And so they chop it up into little bits of, say, 10 or 20 or I'm not sure the exact number of possibilities, but
because you have no idea what you're talking about.
And so then you say, now computer, arrange that into a full genome.
They did not do sequencing. They did assembly.
That is sequencing.
Sequencing is a totally different thing. You don't snip it up. You start with what you assume is an intact genome, an intact strand of RNA or DNA in something. You don't snip it up. You don't do this metatranscriptome stuff from jillions of things. You take what you've got there, and you do what's originally called Sanger sequencing, which you start with the whole thing and you just snip off
Sanger
the
sequencing
nucleotides
is
one at a time.
Not.
And then you listen.
Sanger sequencing does not sequence an entire genome in one read.
Then that's how they do the sequencing for that mouse genome, for the human genome. That's sequencing.
Next-gen sequencing is Sanger sequencing automated. Sanger sequencing, when people sequenced stuff back in the day, before next-gen sequencing, they would do Sanger sequencing by hand and basically assemble a genome piece by piece. It's the same concept. It's just scaled up and automated with next-gen sequencing. And he doesn't understand that because he doesn't actually do any of this. It's wild. He's just about to start a cult with his birds loudly chirping over his sermons.
Where you get the authentic, not random, not scrambled, but you get the authentic sequence as it is, intact.
Oh yeah, we can do that though.
Remember with the SARS, they have all these little bits and pieces of RNA and they stitch them together to try to get a complete sequence. That's what they do. And you don't know where it's coming from, they just know what it ought to look like.
And they keep going with...
Music to my ears. Okay, so here we have this paper. The Architecture of SARS-CoV-2 Transcriptome. So this paper uses a technique called nanopore sequencing. Nanopore sequencing does not require you to do Sanger sequencing, which chops up the DNA into smaller pieces and then sequences them and assembles all of the reads into a continuous sequence. It just sequences the entire thing as it is. So in this paper, there were reads that were almost completely as long as the SARS-CoV-2 genome, encapsulating all of the protein coding regions in a single read. Because nanopore sequencing, let me just bring up a diagram on how this works. So with nanopore sequencing, I got to work on this for one summer, so I'm familiar with at least the concept. So you have an ionic current that flows through a pore. The pore can be of biological material or synthetic material, doesn't really matter. And as the ionic current flows through, you get a trace of current. And then when a molecule like DNA starts flowing through that pore, I can't really zoom in on it, but one molecule at a time, one strand at a time, it starts flowing through that pore. It will block the ionic current. And as it travels through, each nucleotide base is a different size. And each different size of nucleotide base is going to block the pore a different amount. And that's going to translate to a characteristic shape of the ionic flow through your sensor. And as the molecule flows through and all the different bases are blocking the current at different rates, then you can get a sequential trace of which nucleotides are passing through the pore at a specific sequence. And so you don't need to do next-gen sequencing techniques where you chop up the DNA and sequence it in pieces. You can sequence an entire or a very, very long piece of genetic material all at once. And that's what this paper did. It sequenced practically the whole genome in a single read. And I would love to see what virus deniers say about it. But they won't, because they don't want to talk to me. They would rather people like Kaufman, people like Andrew Kaufman and Tom Cowan, they would rather try to do DMCA takedowns of my videos instead of actually talking to me or addressing my stuff. Anyway, let's see what other crap they have to say about sequencing.
This next generation sequencing, where they can play around a bit, because next generation sequencing is a kind of they call it unbiased, meaning they sequence anything that is there. Those 30 billion particles, they sequence all of it.
Yeah.
By doing that, they can still have a leeway where they can say they can play around and claim that there's some kind of virus.
And what they are doing with the genetic material, as they never were able to isolate a piece, even not the whole piece. In case of coronavirus, 30,000 building blocks called nucleotides. Never ever.
Oh, okay.
Even it's standard.
I thought he said 30,000 billion blocks. He said building blocks. Okay. Whew. Whew. Okay.
Technique since decades to show that you have a long nucleic acid present. Then you pick it up, you sequence it, and you repeat it, you always came up with the same thing.
Yep.
They never were able to. What they are doing, they use very short sequences, which they found in their dying cells, and add them up in mentally. And then at the end, you have a mental construct which has no base in reality. No base.
This is called, as they say, this is an in silico genome. In silico means in the computer. So it's a theoretical genome.
It's not theoretical, it's empirically measured.
It doesn't exist anywhere in the sample. There is no 30,000 sequence that's a viral genome. There is no such sequence like that in the mixture.
I just showed it.
None.
I just showed you.
So that's the basis of saying this, quote, virus with this genome does not exist in the biology.
Here it is.
It doesn't exist in the sample.
Here it
It
is.
is created by the computer.
This paper here.
That's what we call an in silico genome.
Sequences the whole thing.
They shared that sequence of RNA
pretty much one go
around the world. Other people around the world now knew what to look for. So now they're starting to. Everybody else is getting samples from people, and then coming up with their own little sequence of a coronavirus. And I think it was as of this past December or so, or something like that. There's this database in Germany that had already cataloged 400,000 different unique sequences of SARS-CoV-2. 400,000 of these things. All right. And not one of them had actually ever gotten a virus. They all do the same thing using genetic transcription. They stitch together all this RNA. The reason they have so many of these things, they don't have the virus.
There have been about 15 million SARS-CoV-2 sequences submitted worldwide. And they all got pretty much the same virus.
So what's really happening when they have 2 million different sequences of this virus? What's really happening? Can you explain to people what does that really mean?
Oh, it just means that precisely because there isn't like a real virus, a real organism, right? Because if there were a real organism, you'd be bound by it, right?
What?
You can't invent one. It's there. E. coli, it's E. coli. You can't produce 2 million different E. colis, right? Because everybody would say,
yeah, you can.
That's E. coli. That's whatever you do different. It's not E. coli, right? But since there is no virus,
there's
real
so
virus, anybody can set up their own version of it.
There's so many different strains of E. coli, dude. So many. And even from person to person, if you isolate their E. coli from their gut, they're going to have SNPs, single nucleotide polymorphisms. They're going to be a little different. It's the same thing. Like, viruses just do the same thing, essentially, as bacteria or other cells, just as viruses. The same principles apply.
With no limitation. There's no gold standard, as you say, right? So everybody can take the sequence, change a couple of letters, and produce a different sequence. And they say, oh, this is the sequence that we did in Rome, or this is the sequence that we did in New York, this is the sequence that we did in London, that we did in India.
I'll just have
That's
this.
how variants are born. But the thing is, of variants, I also published an article on variants, is so ridiculous that I actually developed the concept of the ridiculous science, the scientific ridiculous, because it's such a ridiculous story that it's unbelievable. And no one ever says, the king is naked. No one says it.
Their analysis raises 1,000 questions. If there is no proof that anyone ever isolated a virus, nor photographed a virus.
All right, so we're getting to about 48 minutes in. It's about 10:21 p.m. where I am. So we'll go a little bit longer, but I'm definitely not going to watch this whole thing because it's this first part. This is part 1 of this whole documentary series, and it's 2 hours and 19 minutes. There are 7 parts? No, the whole thing together is 7 hours. I forget how many parts there are. They're like 5 or something. But Jesus Christ, it's so much garbage, and it just gets it gets really repetitive already. So
virus, nor sequenced a virus. How did
the point is we'll see. I'll go like a little bit longer, but I'm not going to watch the whole thing. We're going to end like definitely before 11 p.m. my time.
They make a test for it. Why did people get sick? What were they getting sick from? Was
the
this
virus.
A conspiracy or just a broken intellectual paradigm?
Nope.
The doctors and scientists argue that understanding the test is the key to unlocking the story.
Here we go.
The test that was used to label the public as having COVID, without ever having been validated to prove it could do that.
It definitely did.
The alleged epidemic was caused by the spreading of the test.
The German Drosten paper where they actually built the first swab test to test for the SARS-CoV-2, where they themselves, and that was published at the end of February.
I know what's
They
coming.
themselves said that they created this swab test without even having an actual virus available.
There it is. All right, so this claim. So, what happened here? The people who first published in an academic journal a SARS-CoV-2 PCR test. Yes, they did not have isolates available because it was so early in the pandemic that nobody was sharing isolates yet. People weren't shipping isolates of the virus places. You couldn't buy it. But what we had was the genome published publicly online. And what do you need to make a PCR test? You just need the genome. You don't need an isolate of virus. You can have just the genome and use that to design your primers. You can design your forward primer, your reverse primer, your probe, and a positive control ultramer along with your test. All you need is the sequence. And you can make all of those things that you need to design and validate a PCR test. And that's what they did. And it was later validated by regulatory bodies, by CLIA certified testing labs, all independently in-house to actually detect SARS-CoV-2 at very high rates of sensitivity and specificity. In other words, it doesn't detect flu, it doesn't detect enteroviruses, it doesn't detect Coxsackie viruses, it doesn't detect noroviruses, it doesn't even detect SARS-CoV-1. It only detects SARS-CoV-2.
And so it was created on a sequence sent to them by the Chinese.
SARS-CoV-2, the virus the so-called virus that's never been purified.
Who is this
properly
guy?
Isolated.
Never seen him before.
Identified through PCR testing, and it's a fraudulent medical diagnostic. It was never invented to be a medical diagnostic.
PCR cannot detect infection. It cannot detect the presence of a virus.
We falsified
Let's see.
PCR tests
Let's see.
in a huge paper in December 2020. But the authorities have chosen not to ignore that. The scientific authorities.
No, what happened was a bunch of... All right.
In a huge...
What happened was a bunch of these losers tried to write a letter to Eurosurveillance, which is the journal that published this paper. And they tried to criticize it and ask the journal to take it down. The journal responded to their critique and said, no, we had an independent panel of experts review your letter, and they say no.
Paper in December 2020. But the authorities have chosen...
I made a video on this whole thing, by the way.
Not to ignore that. The scientific authorities have chosen to ignore it. You can read how Peter Borger, molecular biologist, deconstructed with 21 other scientists the whole of the PCR test. It should have been immediately withdrawn when our paper was published. The Eurosurveillance refused to withdraw the original paper.
Because it was stupid. Oh, God. Alright, while that's rolling, let's just look at this. This is the patent that Carrie Mullis submitted for PCR, where it specifically states that various infectious diseases can be diagnosed by the presence in clinical samples of specific DNA sequences characteristic to the causative microorganism. Perfect timing with the music.
Because this isn't about the science. COVID-19, SARS-CoV-2 is nothing to do with science. Otherwise, it would have all been debunked months ago.
There is no virus there. The CDC admitted last December they don't have any SARS-CoV-2 to compare with the PCR test to see if that's what it's actually detecting.
So
yeah, that's
there's not
early
even
in the
a way
pandemic.
to calculate an error. They're
still calling it 2019 nCoV. Those screenshots are literally before it was renamed to COVID-19, which was fairly early in the pandemic.
Talk in the news about false positives and stuff. No one knows the false positive rate. You can't calculate
Yeah, we do.
Because there's nothing.
Yeah, we do. It's very, 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 very low. A false positive would be if a PCR test said that SARS-CoV-2 was present when it's actually not present. That would be a false positive. And we know that it rarely ever does that. And when it does, it's most often because of human error. Either improper sample handling or contamination during the assay. Validated and certified labs that do these COVID PCR assays, the false positive rate is extremely low.
To compare it to because there's no virus. So what you have here is basically a roulette wheel.
Even the WHO realizes that the PCR test for SARS-CoV-2 is really riddled with false positives, most positive and false positive. And from my perspective, it can't be relied on at all. You don't know what a true positive is.
Yeah, we do.
With these PCR tests, there's no way to know. So they're all false positives, all positive and false positive with PCR tests. They're totally useless.
The test is a polymerase chain reaction test.
Mm-hmm. You know what those words are.
So it tests for bits of genetic material that we're told are
unique to the virus.
SARS-CoV-2.
Definitely unique
What's
to the
wrong
virus.
with that is that they assumed it exists through a computer model, as a sequence that was modelled, not a live, a derivative, a sequence derived from, a live isolate.
You have to have a reference standard. For example,
yeah, we have that.
Like I said, the CDC doesn't even have any authentic SARS-CoV-2 to validate the PCR test.
Yeah, they do.
Now, when you've got a PCR test, everybody knows you've got amplification cycles, right?
Mm-hmm.
The more times you run the test, the more cycles you do, the more likelihood of, the more probability there is of a positive outcome, positive test result.
In the test they are using, they are extremely unbelievable, far away from any biochemical reason. They are using 45 cycles. I mean, that's another argument. It's completely out of reason. With 30 cycles already, the PCR...
I mean, look at this screenshot. It's showing different... Not all labs do 45 cycles. These two labs are doing 45 cycles with their kit. And these are doing 40, 38, 35. LabCorp is one of the biggest clinical testing labs in the United States. They do 35 cycles. Are they who's the editor for this documentary? They're not even paying attention to their own claims. Jesus. Okay. So let's see. This is what a lot of these people don't understand. So this is a test run for 55 cycles. Right? This is a qPCR test run for 55 cycles. This sample was the only one that had HPV. It was a positive control. All these samples are negative. At 55 cycles, they're all still negative. And this positive control came up positive around cycle 30. This is what qPCR looks like. It's real time. People don't understand that.
Always starts to be dirty to produce all kind of genetic material, means to give a positive result. Because the enzymes used, they got old with every cycle, heated up, cooled down, so they are not specific. They just produce, even without a given DNA, they just produce genetic material.
The enzyme is not what's specific in a PCR. What determines the specificity in a PCR are the primers. The forward primer, the reverse primer, and the probe in the case of qPCR. It's not the enzyme. The enzyme, Jesus Christ. The enzyme is just recognizing double-stranded DNA that is primed for replication. That's all it's recognizing. It's the primers that determine specificity. This guy has no clue what he's talking about.
Positive result. So therefore, you can test everything and everybody positive, even the most pure water or vodka.
They're basically just saying, hey, healthy people, take this test, right? So we can label you as, a case because we're using that to drive all the policies.
No. So what happens when, okay, here we go. All right, we'll get to this and then we'll probably wind down. I don't know what comes after this, but yeah. Okay, so if someone tests positive for SARS-CoV-2, what's next, right? We know from validated PCR tests that if it's positive and there's no signs of contamination, no signs of mishandling the samples, nothing like that. It's a positive sample. SARS-CoV-2 is there. So what happens next? Well, it might mean that you're going to get sick. Maybe. We don't know when in the phase of illness, phase of infection you are. You could get sick tomorrow. You could get sick a few days after you test positive. You might not feel sick. You might not feel symptoms at all. You might be an asymptomatic carrier. Either way, you can still pass it on to other people. If you're asymptomatic, you can shed enough virus to pass on to someone else. If you're sick, if you're going to get sick, you might want to know that you're going to get sick. Either way, you should act as if you could be spreading virus to other infected people. That is the main point of COVID PCR tests, to know who is infected and where they are, so that people can behave accordingly and act in ways that lessen their chances of spreading it to other people. Now, this guy, the former president of Tanzania, who died probably of COVID, is going to run COVID PCR tests, or claim to have run COVID PCR tests on samples that could not possibly be infected with SARS-CoV-2. Is he telling the truth? Probably not.
Mbuzi, tukachukua sample za kondoo, tukachukua sample za papai, tukachukua sample za oili ya gari na sample za vitu vingine mbalimbali, tukavipeleka pale kwenye maabara bila wao kujua. Ya papai tukaipa jina Elizabeth Anne, miaka 26, female. Papai lile lilikuwa positive kwamba lina corona.
Sure it did.
Maana yake maji mle ndani zilizotolewa mle kwenye papai ni positive.
This guy, by the way, pretty much denied that COVID was a problem, and he ended up passing away probably due to coronavirus. We don't know, but it's possible. And I should clarify with what I was saying about COVID PCR earlier. The only way you would know that you are not infectious would be to do a series of experiments that are impractical to do on a population level, because you would have to determine what is the infectious titer of whatever material the patient is shedding during their infection, and whether or not that infectious titer can be correlated to actual transmission. By the time you do all those tests, it's too late. It takes too long. It takes too long to do those assays to determine how infectious a material is. By the time you've finished, they've already infected other people if they're walking around as normal. So that's why when you test positive for PCR, you have some sense of responsibility to act like a functioning member of society and not put other people at risk. That was the point during the pandemic.
Tulipeleka sample ya ndege Kwale, imekuwa positive. Kwa hiyo ukishagundua kitu hiki, lazima ujue kuna mchezo wa ajabu unafanyika kwenye vipimo hivi.
It's a bit like fish. It is sad that he passed away. I mean, if it's true that if he did truly die of COVID, then, you know, he's a victim of science denial, and that's genuinely sad. All right, we're gonna go for like maybe another 5 more minutes, and then call it a night.
You know, and that's what it is. All this testing and screening, all this, you know, PhDs, all the rest of it. You know, it's a dragnet for generating markets for drug companies.
So what we see happening is when they do their genomic sequencing, sometimes you detect slightly different sequences from previous.
Because genomes mutate.
Wow, that looks like a new variant. And the most recent one was, Omicron at the moment, and that's. I mean, clearly this was just a way to create more cases, because what they said was that previously you'd have a PCR.
Yes, they detected Omicron, and then every diagnostic lab around the world subsequently decided to jump in on the lie and say, yeah, we're also detecting Omicron. Yep. Everyone's lying. Totally cool worldview.
Yeah, that would look at 2 or 3 specific selected nucleotide sequences. And with Omicron, they said
these people know that
the S gene or the spike protein,
these people know that genomes mutate, right?
Was mutated so that the existing PCRs were not detecting that particular sequence anymore.
Right.
So essentially, what was previously a negative test is now a positive test. I mean, this is how crazy.
No. So what happened was COVID PCR tests usually detect more than one target. So there will be a target, a set of primers and probes for detecting the S gene, the spike gene, and another set of primers and probes for detecting like the E gene or the ORI gene, or the ORF gene, excuse me. And so normally the tests would require all of those targets to come back positive in order for it to be a true positive. But they noticed that a lot of their samples were not coming up positive for spike. And then when they sequenced those samples that weren't coming up positive for spike, they saw that the sequence was different, that it had mutated, as genomes do, as viruses do. And it turned out that was Omicron. Just adding context for whatever garbage this guy's saying.
It's getting. So previously you'd have 2 or 3 of the selected sequences would be called positive, whereas now you don't need one of those sequences to be positive. And
well, then what they do is they redesign the test to be able to detect the spike gene that's specific to Omicron. And then it works.
They say, well, that's because it's Omicron. So it's totally obscene. It's I've never seen anything like it where a previous.
So what do they think the test is detecting? If they have the E gene and the ORF gene coming up positive, but not the S gene, but the S gene was coming up positive, and when all the other variants were circulating, what do they think the test is detecting? What do they think is changing? Do they think something about the human cells that they're sequencing, that are also in the sample, that are being sequenced by these labs, is changing? Like, what do they think is happening? It's so bizarre.
Positive test becomes negative, but then you call it positive because you say that it's because there's a new variant. And that's why we saw an explosion, as predicted, in the number of apparent cases. So at no time did they take a group of people who were said to have this Omicron variant. They didn't study those people to see if they were actually unwell or what happened to them over a course of time. They did none of that. There's no clinical work going on.
Yeah, they did.
It was simply a declaration that we've detected a slightly different genetic sequence, and hey presto, when other places around the world started checking
they literally
genetic
sequenced.
sequence, they start finding it too. But again, you don't need the presence of a virus to do that. These genetic sequences can be found anywhere once you start looking for them.
I'm very interested in
find a SARS-CoV-2 genetic sequence in like just a pure cell culture. You can't do it. You can't do it. Go to frozen stocks of like blood, human blood, or biological samples that were stored before 2020 and try to find SARS-CoV-2. You cannot do it. It doesn't exist.
Recent developments in medicine. So, for instance, I've been following the debate about fats. Recently, I've been following the debate about
what?
COVID-19, trying to figure out what is scientifically valid, what is not, how do the social powers that shape science interact to produce facts. And what I've been observing was that we run the danger of going into another scientific mythology without great caution. That's my opinion. Because if you look at what happened was, initially you have some strange infectiology, some people dying, some this, some that, but without that PCR test that was developed, you would never have been able to say there is a new coronavirus. Like,
you would
let's imagine
be able to say there's a new illness.
A time 20 years ago or 40 years ago where you didn't have the test, right? All you had to go by was...
We had PCR 20 years ago, dude. What? And I mean... Jesus Christ. I mean, just look back to the 2019 flu. People didn't know that was a virus. People didn't know what viruses really were back then. But people knew that a ton of people were dying in excess of what we would be expected. What we would expect. And so, I mean, if we look here, let's see. Let's just do America. Oh, that's the United Kingdom. I don't want the UK. I want United States. There we go. All right. So if we look at this, this is excess death. We would have definitely noticed, even without a PCR test, that a lot more people were dying than normal. This is excess death compared to a 5-year average. This isn't normal. A lot of people were dying. We would have noticed this. It doesn't take a PCR test. If you never test for cancer in your lifetime, it doesn't mean that cancer is not going to kill you.
Was patients who go to hospital, patients who are severely ill, and patients who die.
Mm-hmm.
If you use those indicators, at least in Germany, you would have never seen any exceptional way.
Oh, really? Really now? Let's see. Really? No one would have noticed. No one would have noticed 36% spikes in excess deaths. No one would have noticed this? Really?
Ridiculous. Of any infection, because there was none. Hospitals were not more full or empty than they were during the time of winter infections. You had not more people dying. You would not have known anything. So part of that corona crisis is actually a testing spree.
You know what? Jesus. I'm gonna probably end it around here. And instead of playing more of this crap, I'll recommend this is an ad right now, but I'll recommend this movie called The First Wave. Documenting obviously the first wave of COVID.
I have to keep it together. I have kids who can't see me fall apart. He has to come home. He has no choice.
And just let my fear be my strength, because I know one day I'm gonna be with my wife and my baby.
It is because of you that we are gonna make it through.
You are more than just a doctor. You do it from the heart.
Every time you see the COVID patient, you can't help but say, damn, this can easily be my mom.
Each one is getting harder and harder.
It's tough to see people constantly have to suffer. When we started chanting, I literally felt like my breath was stripped away. I also heard all the times my patients said, I can't breathe.
Guys, we need some help in here now. Everything perfect.
Wait, stop. I have a pulse. I have a pulse right here. Pulse.
What we've been doing here over the last number of weeks is extraordinary and special. And you are fearful, you are stressed, but you're...
All right, you get the idea. It's a documentary about like what it was actually like at the front lines. I mean, it was obvious that something was happening, even if we have no knowledge of what viruses are. It was obvious that a lot of people were dying at rates way higher than we're used to. These people just deny it. They just deny all of it, and they deny it because they want to push a comforting reality where there's no such thing as viruses that are going to hurt you. All that stuff is fake. Just buy my supplements, buy my DVDs, buy my seminars, and I'll teach you how to be healthy for real. Because viruses, they're all fake. Yeah. So, anyway, it's getting late here. And we're literally not even halfway through the first part of this documentary series. It's god-awful. So, yeah, that was a pretty terrible documentary. Was it worse than Watch the Water? I don't know. Probably not. I think Watch the Water was definitely worse. But I think I'm going to start to wind down here. What's going on in the chat? Anyone have any final words as before we wrap up here? Yeah, this documentary is definitely not as bad as Watch the Water, but it's pretty bad. It had worse editing. Yeah, well, Watch the Water barely had any editing to do because it was just one dude talking the whole time. And talking about his fortune cookie story. And this was fun. I enjoyed doing this. I'll definitely do more of these if you guys want. It was truly horrible, yeah. And the editing was pretty bad. All right. Well, I had one super chat. Bobo Not says, thanks for all your debunking material. Family members' never-ending misinformation was wearing me down. You've informed and given me confidence. Thank you, Bobo Not. I'm glad to have helped. And stay strong in your conversations with your family. I know it can be tough. Anyway, yeah, it's pretty late for me here. I'm probably going to go to bed. And yeah, that documentary was terrible. Classic, just same old virus denier tropes. I can't imagine how they stretch the same claims over and over again into like 7 hours. Just milking it for all it's worth. All right. Next week, I hope to. Next week, I'm gonna have a short video about one of John Campbell's recent videos, his 1 in 35 garbage. After that, I am. Oh, apparently this Zoth guy's been talking a lot. How do I sleep when I know I'm spreading lies for corporate profits? I'm not. Literally not. Vaccines make the least amount of money of any pharmaceutical drugs. They make the least amount of money because you need just like 3 to 5 doses of most vaccines, and that's it. Whereas a drug like, say, ivermectin, if that had worked for COVID, people would be getting prescribed it every single time they test positive. That would have made a whole lot more money. So, no, I'm not. And I sleep just fine. Until my daughter wakes me up in the morning. Anyway. So, what was I saying before? Oh yeah. After I make that short video, I'm probably going to have more guests on my channel. Because my second child is coming up soon, this fall. And at that point, it's going to be harder for me to make regular weekly videos, obviously, so I want to try to have some content built up in the form of guest conversations. Anyway, that's my plan. So hope you all enjoyed this one. I'll do more of these in the future. My patrons voted on this documentary. There will be other choices in future polls. So, yeah, it'll be fun. And we'll see if a documentary is as bad as Watch the Water. But anyway, yeah, this will be my second kid, so double the work. Enjoy your night, everyone. I'll see you next time. Thanks for tuning in. This was fun. I enjoy doing these, and I'll see you next time. All right, take care, all. Thanks. Bye.